This is week 8, genetic variation in populations. The genes that are responsible for the traits that Mendel studied when we were looking. This slide illustrates inheritance of linked genes. In looking at this slide, we see that it's divided up into part A and in recombination is where the chromosomes This slide shows the crossing over disrupts linkage. This slide shows that the allele configuration is important. Crossing over is more likely to occur between. In this slide, we're looking at linkage versus non-linkage. The frequency of a crossover between any two linked genes. To illustrate how to determine the degree of linkage by percent. Okay, let's look at Greg and Susan. Both have the Nail-Patella syndrome. Let's look at Susan. Susan has type B blood. She can either be BB or BO. Susan's mother has Nail-Patella syndrome and type O blood. Susan's father has normal nails and type B blood. Therefore, Susan's mother's blood type is OO. Susan's father is either BB or BO. Her alleles are trans because we see that Susan got the dominant allele B from her father, and her blood type is actually BO. Her genotype is BO. And Susan's mother gave her the Nail-Patella syndrome gene. Therefore, Susan's alleles are in trans. Okay, this slide is showing you the inheritance of the Nail-Patella syndrome. So we now looked over what were the genotypes of Greg and Susan, and we looked at their parents. And we're going to look at a question that Greg and Susan have asked the genetic counselor. They have asked the genetic counselor, what is the possibility, what's the chance that their child inherits normal nails and knees and type O blood? The genetic counselor deduces their allele configurations using the information about the couple's parents. The information that we had, we were able to use this information to be able to figure out what would be the possibility that they could have a child that they have just asked about. The only way that this genotype can arise from them is if the sperm do not carry the Nail-Patella gene, which occurs with a frequency of about 45% based on pooled data, and that this sperm fertilizes an oocyte, which carries the O allele, which occurs 5% of the time. The result, according to the product rule, is a 2.25% chance of producing a child that has normal nails and knees and type O blood. So please look over this chart for a few moments. I will pause it for a few moments so that you can look it over and go over the information again. A linkage map begins to emerge when percent recombination is known between all possible pairs of 3 or more linked genes, just as like a road map. With more landmarks, you provide more information on distance and direction. So let's consider the genes X, Y, and Z. If the percent recombination between X and Y is 10, between X and Z is 4, and between Z and Y is 6, then the order of the genes on the chromosome is X, Z, Y, the only order that accounts for the percent recombination data. It is a little bit like deriving a geographical map from knowing the distances between the cities. Genetic maps derived from percent recombination between linked genes accurately reflect the order of the genes on the chromosome, but the distances are estimates because crossing over is not equally likely across the genome. Some DNA sequences are nearly always inherited together, like two inseparable friends. This non-random association between DNA sequences is called linkage disequilibrium, LD. The human genome consists of many LD blocks, where stretches of alleles stick together or interspersed with areas where crossing over is prevalent. The first human genes mapped to their chromosomes encoded blood proteins. Because these were easy to study, in 1980 researchers began using DNA sequences near genes of interest as landmarks called genetic markers. These markers need not encode proteins that cause a phenotype. They might be DNA sequence differences that alter where a DNA cutting enzyme cuts, differing numbers of short repeated sequences of DNA with no obvious function, or single sites where the base varies among individuals. The term genetic marker is used popularly to mean any DNA sequence that is associated with a phenotype, usually one affecting health. Computers tally how often genes and their markers are inherited together. The tightness of linkage between a marker and a gene of interest is represented as an LOD score. If we look at an LOD score of 3 or greater, it signifies linkage. It means that the observed data are 1,010 cubed times more likely to have occurred if the two DNA sequences are linked than if they reside on different chromosomes and just happen to often be inherited together by chance. It is somewhat like deciding whether two coins tossed together 1,000 times always come up both heads or both tails by chance or because they are taped together side by side in that position as are linked genes. If the coins land with the same sides up in all 1,000 trials, it indicates that they are likely taped. Before sequencing of the first human genomes, genetic markers were used to predict which individuals in some families were most likely to have inherited a particular disease before the symptoms began. Such indirect analysis are no longer necessary because tests directly detect disease-causing genes. However, genetic markers are still used to distinguish parts of chromosomes. A haplotype is a set of DNA sequences inherited on one chromosome due to linkage disequilibrium. It makes it possible to track specific chromosome segments and pedigrees, and disruptions of a marker sequence indicate crossover sites. In the next slide, you're going to be able to see how it is designated in a pedigree. And the numbers in the bars beneath the pedigree symbols enable researchers to track specific chromosome segments with markers, and disruptions then would as I said, would indicate that there was some crossover. And when we take a look at this, we can see what I'm talking about with that. So knowing the haplotype of the individual 2,2 reveals which chromosome in parent 1,1 contributed the mutant allele. Because Mr. 2,2 received haplotype 3,2,3,3 from his affected mother, his other haplotype 2,2,2,2 came from his father. Because Mr. 2,2 is affected and his father is not, the father must be a heterozygote, and 2,2,2,2 must be the haplotype linked to the mutant CFTR allele. Today, entire genomes can be sequenced in under a day. However, researchers are still filling in the orders of the genes on the chromosomes and determining gene functions and interactions. This will end what we will do for week 8, and we will begin on January 2nd with week 9. Have a great holiday.